Hello everyone, welcome to Sankalp Learning Solution. So today we are going to solve the LBA-KCET questions on fifth chapter, that is molecular basis of inheritance. So let's begin with the topics. So today we are going to discuss about the structure of DNA and packaging of DNA helix and the search of genetic material and properties of genetic material, and then third RNA world and the replication process, and then we also discussing transcription and then transcriptional unit and the genes, and then types of RNA and the process of transcription, and then sixth genetic code and then mutation process, and then seven translation and tRNA, and then regulation of gene expression and the lack operon process in the prokaryotes, and then human genome project and the goals of human genome project, and then lastly we are going to discuss about the DNA fingerprinting. Okay, so these are the topics we are covering in the molecular basis of inheritance. So let's begin with the first question. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 1. The bond formed between the deoxyribose sugar of one nucleotide and the phosphate group of next is. So dear student, so just understand the question properly. The bond formed between the deoxyribose sugar and the nucleotide they are asking. Okay, so let me explain properly here, because here you should know first what are the components of DNA. So let so dear student, there are three components present in the DNA. First one is a pentose sugar, and then second one phosphate group, and then third one, so that is nucleic acids, or you can also call nitrogen bases. Okay, so these three are the components present in the DNA. If you observe here, so here this one is phosphate group, and here it is pentose sugar, and here it is nitrogen base. Okay, so this is what three components present in the what DNA. So here let me explain the pentose sugar present in the DNA is deoxyribose. Okay, and the structure is here. This is how it looks. All this why I am explaining is for understanding the bond present between this, understanding structure is really necessary. So this is what the structure of pentose sugar. So here it is containing C5H10O4. So this is the molecular formula. Okay, and then phosphate group is present here, right? So the phosphate group will be like this. Okay, okay. And then here one more phosphate group is present to the another ribose sugar, right? Another deoxyribose sugar. Here one more phosphate group is present. So to explain the bond, so I am writing both the phosphate group here. So on the both sides of the pentose sugar, it is at connecting to the phosphate groups. Okay. So here, if you observe, dehydration reaction is taking place due to the dehydration reaction. Okay. So here, if you observe H2O, so here H2O will be removed and it forms the ester bond here. It is going to form ester bond. Okay. And here also the third position of the pentose sugar, you observe here also dehydration reaction and forms ester bond. Okay. To the fifth position of pentose sugar and then third position of pentose sugar. So here phosphate groups are attaching and forming the ester bond. Okay. So here phosphate group, that is phospho. Ester because here it is forming the ester bond. Diester because here two sides, to the fifth position as well as to the third position, it is forming ester bond. So diester bond. So in between the deoxy sugar of one nucleotide and the phosphate group of the next. Okay, so here it is having the bond that is called phosphodiester bond. Phosphodiester bond. Okay, I think now you have clearly understood the topic. So the correct answer is here, dear student. Which one? If you observe properly all the options, so here so many bonds are given here in the option. So the correct answer is option C, that is what phosphodiester bond is present. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question second. Which DNA region has the highest melting temperature? So dear students, so here if you observe the DNA structure. Okay. So DNA has two strands, right? DNA containing strands. Right. So both the strands are helically wounded each other to form DNA helix structure. So this is called DNA helix structure. Okay. So this is what the two strands are anti-parallel to each other. 5 dash to 3 dash. 3 dash here 5 dash to 3 dash. Okay. So here there will be a nitrogen bases in between the two strands, right? So nitrogen bases are connecting like this. Okay. So these nitrogen bases are held by the bond that is called hydrogen bond. If you observe here, so this is what the hydrogen bond. In between the adenine and thymine, there is a two hydrogen bonds, and in between the guanine and cytosine there will be a three hydrogen bonds. Okay, so you only just think about this. So which bond will be having highest melting temperature? So here it has three bonds, so of course three bonds will be having highest melting temperature. So the GC region has the highest melting temperature because here three hydrogen bonds are present and it is more stable. Okay, so that's why here it needs highest melting temperature. Okay, so the correct answer is here, dear student. Which one? The option B, that is what? 50% GC rich region. Okay, 20% will be very less. So the 50% is the correct answer. That is what GC rich region. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question. C. The distance between two adjacent base pair in a bDNA is. So dear students, so bDNA, this is what the structure of human DNA. So that is what bDNA. If you observe here, these are the base pairs, right? These are the base pairs, nucleotide base pairs. And here if you observe, so 3.4 nanometer, that is what here it is a pitch. This is called pitch. Okay, this pitch is called what? The distance of pitch will be 3.4 nanometer. And if you observe here, this one is called major groove, and then this region is called minor groove, and the basis, and in between the basis, hydrogen bonds are present, and the width of the DNA is 20 nanometer. Okay, 20 nanometer. So here they are asking the distance between the adjacent base pairs. So here these are adjacent base pairs. Okay. So the adjacent base pairs will be having 0.34 nanometer distance. Okay. And for each turn it will be containing. This is one turn. Okay. For each turn this will be containing 10 base pairs. Okay. So this is about the DNA structure. So the correct answer is here, dear student. Which option? Option B, that is 0.34. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 4. Assertion. DNA replication is semi-conservative. Reason. Each new DNA molecule contains one complement new double strand. Dear student, please read the both assertion and reason properly. So DNA assertion is DNA replication is semi-conservative. Is it correct? Yes. The DNA replication is called semi-conservative process. Why it is semi-conservative process? See, what do you mean by DNA replication? Replication is a process of formation of exact copy of the parent, right? So that is called what? Replication process. Okay. So here this is the DNA strand. So here this will be separated, right? So this is called template strand, because here in the replication process both the strands of the DNA act as a template strand. Template strand means here these are non-coding strands, or it also called antisense strand. Okay, so both the strands are acting as a template strand. Okay, according to this template strand, newly one more strand is synthesized here. So that is what we called a replication process. So this newly synthesized strand and template strand together becomes DNA. So this DNA will be similar to the parental strand. Okay. So as you know that here it is template strand. According to the template strand here, the complementary strand will be synthesized. Okay. So that's why here the DNA replication is called semi-conservative. Okay. So the assertion is correct. But reason, each new DNA molecule contains one complete new strand. See, this is the new DNA molecule, yes. But is it is containing both the new strand? No. Here one strand is older one only. That is what template strand. So to the template strand, the whatever the complementary strand is there, that is only the new one. Okay, not both the strand. So the reason is not correct here. So which one is correct answer, dear student? The option A, B, that is A true and R is false. Okay. So the assertion is correct here. Reason is wrong. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 5. The histone that does not form a part of nucleosome core is. So dear students, so this is what the histone. Okay. So histone means what? So there will be a positive proteins that is called histone proteins. Histone proteins. Okay. So these are what? Positively charged. Okay. So our DNA is what? Negatively charged. Right. DNA is negatively charged. Right. So this our DNA will be surrounded surrounding or surrounding this what octamer nucleosome, octamer nucleosome. So this structure is called what? Whole structure is called nucleosome structure. Okay, and they are asking here which part is not belongs to the nucleosome here. Okay, so here there will be one more histone that is called H1. Okay, so that is linked to this nucleosome, and here the one more nucleosome it is connecting. So that's why it is called linker DNA. Linker linker protein. Sorry, linker protein. That is what H1. So here H1 protein act as a what linker, linking DNA. So that's why here the correct answer is which does not belongs to the part of nucleosome is the option A, that is H1. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 6. Which enzyme removes RNA primers during the replication? So, dear students, already you know that what do you mean by replication? So, replication is a process of formation of exact copy of another parent, exact copy that is similar to the parent. Okay, so that is what the replication process. So here during the replication process, so here as you know that DNA cannot synthesize from the 3 dash to 5 dash. It should always start from the 5 dash to 3 dash, right? So here DNA, there are three main enzymes: DNA polymerase 1 and DNA polymerase 2 and DNA polymerase 3. Okay, so these three enzymes which are involved in the replication process, and the DNA polymerase 1. So here this removes the primers after replication process. Okay, and then DNA polymerase 2 that cross checks the process. Okay. And DNA polymerase 3, which is adding the primers for initiating the process of replication replication. Okay. And then it also adds nucleotides nucleotides. Okay, complementary nucleotides. So these three enzymes are involved in the process. Okay, so I have told you the functions also now, right? So just read the question now. So the which enzyme removes the RNA primer, they are asking. So which enzyme is removing here RNA primer? So here the DNA polymerase 1. So that is removing the RNA primer. If you observe here, this is the RNA primer. So that is removed by the DNA polymerase 1. Okay. So the correct answer is here option B, that is DNA polymerase 1, that is removing the RNA primers. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 7. Statement 1. RNA polymerase 2 synthesizes the mRNA in eukaryotes. Statement 2. It binds to the 10 a minus 10 TAT AAT region. So dear students, so let's solve this question. So the statement first, RNA polymerase 2 synthesizing the mRNA. So what this process? So synthesizing the mRNA from the DNA strand. Synthesizing mRNA from DNA. Okay. So that process is called what? Transcription process. Transcription process. Okay. So in this process, okay, again there are three enzymes are involved. So RNA polymerase 1, RNA polymerase 2, and then RNA polymerase 3. Okay. So here RNA polymerase, so that is involved. So RNA polymerase 1 is involved in the synthesizing 28 sRNA, 18S and 5.8 sRNA. And then RNA polymerase 2, that is involved in the formation of heterogeneous RNA or mRNA. Okay. And RNA polymerase 3 is involved in the formation of small nuclear RNA or 5S rRNA. Okay. So this is what the function of the RNA polymerase 1 and 3. Okay. So you should add this properly, then you can easily solve the question. So here what they are asking, RNA polymerase 2 synthesizing mRNA in eukaryotic. Yes, in case of eukaryotic, so the RNA polymerase 2, so that is involved in the formation of heterogeneous nuclear RNA, or it is also called the matured mRNA, messenger RNA. Okay, so the statement 1 is correct, and it binds to the 10 TATAT region. Is that true? No, because here this is in case of what? Prokaryotes. Okay, but in case of eukaryotes, it's binding to the region where contains 7 base pairs TATA TAT. So this is what the correct region where exactly what this what mRNA RNA polymerase will get bind. Okay, so in case of eukaryotic contains 7 bases, nucleotide bases. Okay, so the statement 2 is not correct. So which one is correct answer, dear student? Now read all the statement. So the statement B, that is 1 is true and the statement 2 is false. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 8. Match the following. So dear students, they have given two column here. In the first column, they have given the process. So here there is a one more extra procedure in the process of transcription. So what do you mean by transcription, dear students? I think you know just a minute back I have explained this. Transcription is synthesis of mRNA from the DNA. Okay, so that is what transcription process. So here, in case of eukaryotes, so here one extra process is there that is called forced transcriptional mechanism. Forced transcriptional mechanism. Okay, so this process involves that is called splicing. Okay, in the splicing, first process is uh, capping, second one poly A tail, okay, and then third one splicing. Okay, so these are the three process okay involved in the forced transcriptional mechanism. Okay, so here in this process, see what is these all the process they have given here. Okay, so first one 5 prime cap. Okay, that means here so there will be a mRNA, right? So here this is not matured now. Okay, so here there is a still it has to undergo the post transcriptional mechanism. So here this one is 5 prime end, this one is 3 prime end, right? So there will be a enzymes that is called exonucleases. Okay, so to protect from these enzymes, so these enzymes what they are doing means they will start cutting the ends of the strands. Okay, so to protect from these exonucleases, so here there is a post transcriptional mechanism that is called capping and then tailing process, and then the remove splicing, that is the removing of exons of introns, and then uh, what ligating the exons, then we will get the matured mRNA. Okay, so this is what the post transcriptional mechanism. So here they have given pi prime cap, that means here to the 5 prime end, okay, 7 methyl guanosine 3 phosphate groups are added to the 5 prime end. Okay, so that process is called what? Capping. Okay, cap is added to the 5 prime end, and to the third prime end here adenine residues are added. 200 to 300 adenine residues are added like this. Okay, so that becomes poly A tail, poly A tail. Okay, and then the splicing, so in between the mRNA strand, there will be a introns. Okay, so those are called non-coding sequences. Non-coding sequences. Okay, so here non-coding sequences has to be removed from the mRNA because mRNA will be containing only coding sequences. Okay, so that's why here the removing of introns and adding the or ligating this what exons that is called splicing. Okay, so these are what three different steps involved in the post transcriptional mechanism. So now you can easily match, right? So 5 prime cap, that means adding 7 methyl guanosine cap. Okay, and then adenine residues means adding polyetyl to the third prime end. And then exons. So here exons are what? Coding sequences, that means explicit sequences. Introns are non-coding sequence, so that has to be removed from the mRNA. So here these are removed sequences. So which one is correct answer, dear student? So if you observe, so the option A is correct here. The option A is 2, B is 1, C is 3 and D is 4. Thank you. Hello everyone. Welcome to the Sankalp Learning Solution. Let's solve the question 9. The anticodon is located on. So dear students, so in the RNA there are three types. One is mRNA, rRNA and then tRNA. Okay. So here mRNA is called messenger RNA. Messenger. rRNA is ribosomal RNA. Okay, and tRNA is transfer RNA. Okay, so here whatever you are observing the tRNA, the picture, so that is called a very smallest RNA, smallest RNA. Okay, so that is containing smallest because that is containing only the single strand and which is folded like this. Okay, so that will be containing at the 5 prime and hydrogen bonds, at the third prime and amino acid attachment site is there, and there will be a two loops, a DNA loop, a D loop and the T loop, and there will be a variable loop, and then here it is containing the three triplet codon, that is called anticodons. Okay, so this anticodon will be coding with the mRNA sequence of coding region. So it forms what? Amino acid, particular amino acid. So that will become the protein. Okay, so this is what the function of tRNA. So here anticodon is located in the what? The answer is which one? The smallest RNA. So that is what? tRNA. So the answer B is correct here. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 10. Which of the following is a stop codon? So dear students, so just observe the image here. So this is what representation of codons, 64 codons. Okay, so there are 20 amino acids, 20 amino acids, okay, coding 64 codons. Okay, so these codons are triplet in nature. Okay, and writing the codon is very easy. So if you observe here, UCAG, these are what the bases, you uracil, cytosine, and then adenine, and then guanosine. So like this, on both the sides you should write. Okay, and then each uh, what column will be containing four codons here. So like this you can pair U and you should write U. Okay, in the end a U will come, and U and then in the end C will come, and then again you are pairing U and the end A will come, and again U and then G will come. So like this the writing the codon is very easy. Okay, and there are three stop codons. So that is called UAA, UAG, and UGA. Okay, the remembering the stop codon is little complicated only. Definitely you will get confused. So you buy all the stop codon UAA, UAG, and UGA. Okay, and then there is one start codon that is called AUG. Remembering start codon is very easy, but the stop codon is little confusing one. So by this UAA, UAG, and UGA are stop codon. So which is, so here there are four options they have given. Which one is stop codon now? So the UAG. So the option C is stop codon. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 11. In a diagram of tRNA clover leaf, the amino acid attachment site is located on. So in the last question only we discussed the structure of tRNA, that is the smallest RNA, right? So here this is the third fragment, that is what amino acid attachment site, right? And here it is kilo, and this one is variable arm. Okay, so here these three are what anticodons. Okay, and here it is D loop, and here it is a 5 prime end. So that is what hydrogen bonding site, hydrogen bond site. Okay, so this is what the structure of and labeling of the tRNA. Okay, so here they are asking, so in the clover leaf of tRNA, amino acid attachment site. So the correct answer is here not a 5 prime end, that is hydrogen binding site. Anticodon arm is also not correct. Variable arm is also not correct. So the third prime CCA is a codon, end codon of the third prime. So that is what amino acid attachment site. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 12. In prokaryotes, The lac operon is switched on when. So dear student, so here lac operon is a process. Okay, it is a process. Okay, where the three structural genes are involved in the synthesizing the enzymes. Okay, and that enzymes are involved in breaking the lactose and transporting the lactose. Okay, so this is what the lac operon process. Okay, when this lac operon process gets activated is the question, right? So here it is in case of bacteria. Okay, bacteria, that is what in case of prokaryotes. So in bacteria, if the glucose is available, so the bacteria are taking absorbing directly glucose for the energy. Okay, so if the glucose is not available, so in that case, so whatever the lactose is present in the atmosphere or the surrounding, okay, so they will start absorbing the lactose. For that, it will switch on the mechanism that is called lac operon. Lac operon. Okay, so here, so the correct answer is here when the glucose is absent and when the lactose is available. Okay, in that case, what this lac operon gets switch on. Okay, so the correct answer is here answer C when lactose is present and the glucose is absent. Okay, so thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 13. Which component of RNA polymerase in prokaryotes helps in promoter recognition? So dear student, so they are asking the RNA polymerase in prokaryotic helps in recognition of promoter region. So this is in case of the transcription process. Okay, so you know the meaning of transcription. That is what synthesis of mRNA from the DNA, right? From the DNA, they are synthesizing the mRNA, right? So that is what transcription process. So in the transcription process, you know that already I have studied and you have also solved the question on this topic. So the RNA polymerase, three RNA polymerase are involved, right? So in that, the RNA polymerase in the prokaryotic. So which region of RNA polymerase so helps to recognize the promoter sequence they are asking. So this is the DNA, prokaryotic DNA, right? So here it has to recognize the promoter, okay, promoter region. So here RNA polymerase will be containing the two factors. One is a sigma factor, another one is RHO factor. Okay, so these both the factors helping. So alpha factor will be helping for recognition of the promoter region and RHO factor will help to release the mRNA. Okay, so this is what the function of what the factors which is a region which is present in the what RNA polymerase. So the correct answer is here. Which one, dear student? The option A is also not correct. B is also not correct. Even D. So the correct answer is here. Answer C. That is what the sigma factor which helps to recognize the promoter region of the prokaryotes. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 14. DNA ligase form. So dear students, there are several enzymes involved in the replication process. Okay. So let me explain all the enzymes once. So for you it will be very clear. One is DNA helicase. Okay. So it helps to separate the strand. Okay. And then DNA polymerase. So here a polymerase enzyme which adds nucleotide and primers. Okay. And then ligase. Okay. So ligase enzyme that seals the nucleotides of DNA. Okay. And then SSB, single stranded binding protein, that is, okay, it helps to what? Unwinding process. So unwinding process of what? Strands of DNA. Okay. And then DNA gyrase, it prevents supercoiling. Okay. So these are the different enzymes involved in the process. Okay. So here they are asking DNA ligase. So DNA ligase, what it is doing? So it is sealing the nucleotides. Okay. So that means here, if you observe, there are Okazaki fragments. So after synthesis, it has to the enzyme has to seal here in between the fragments. So that is what done by the ligase enzyme. So if you observe here, it is sealing the two strands here. Okay. So that sorry, two fragments here. So that's why here sealing means what? What it is doing actually. So it is forming the phosphodiester bond. Okay. So already I studied that the phosphodiester bond is present between what? The nucleotide sequence of a phosphate group and the what deoxyribose, right? So that's why it is what forming that bond. So the sealing will be easy. Okay. So the correct answer is answer C, that is phosphodiester bond. Okay. So ligase will be forming phosphodiester bond between the nucleotides. Okay. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 15. A number of base pairs per turn in a BDNA. So as you know that BDNA is a human DNA, right? So I'm not drawing properly. Please adjust, get adjust. So here it is a BDNA, okay? And here this is a per turn. So here 3.4 nanometer, okay, length will be. Okay, so here base pace per turn they are asking. So per turn, so already I have told you the answer. So per turn that they will be containing 10 base pairs like this. Okay, and in between the base pairs there will be a hydrogen bond. Hydrogen bond. Okay, so here 10 base pairs are present for each turn. Okay, so the correct answer is Option C, that is 10 base pairs are present per turn. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 16. If a DNA molecule has 1200 adenine bases, the number of total nucleotide in that molecule is. So dear students, according to Chargaff's rule, what is that Chargaff's rule? Always number of adenine is equals to number of thymine. Okay. And then number of guanine is equals to number of cytosine, right? So that is what Chargaff's rule, right? So here they are giving adenine bases are 1200. So that means what? Thymine also will be 1200. Okay. So that becomes 2400. They are asking the number of total nucleotide. So here we have to guess the number of GC also. So the remaining what here if it is 2400, so we cannot predict directly in what GC. Okay, so here if you think that it is also containing 1200, so here also it will become 1200, so total here also it becomes 2400, so the it will become 4800, the molecule, what number of nucleotide present in the molecule. Okay, so here if they here they haven't, they are not given here what the number of nucleotide present in the GC region. Okay, so the answer, so we have predicted the answer will be a C, that is 4800. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 17th. The region where replication begins in prokaryotes is. So dear students, so they are asking the region where replication begins. Okay, so there will be a region. Okay, so that is called. Okay, so here you know that the helicase enzyme comes and that unwinds the strands of DNA, separates the strands, right? So here, so there, that is the region, exact region where replication start. Okay, so if you see that here it is the region, so that region will be containing same nucleotide, and that region is called origin of replication. Okay, so that region is called what? Origin of replication. So the correct answer is here the option, that is not a promoter, that is not a operator, even it is not an enhancer. So the correct answer is in case of replication, so the origin of replication site will be there. So the correct answer, answer B, that is Ori site. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 18th. Short fragments of DNA synthesized on the lagging strands are called. So dear students, as you know that DNA replication is DNA replication is semi-conservative, right? And here, during the replication, DNA it can only synthesize from 5 dash to 3 dash, not to 3 dash to 5 dash. Okay, so that is not possible. Okay, so here only the way it has to synthesize 5 dash to 3 dash. On both the sides of template strand, it cannot synthesize 5 dash to 3 dash directly. So that's why here it adds us the small fragment so that it will it will make the chance which becomes so 5 dash to 3 dash. Okay, so and that strands, those small strands are called Okazaki fragments. So that is present in lagging strand. So that's why it is called lagging strand. Okay. So the small fragments are called what? The Okazaki fragment, not introns, exons, or initiator fragment. So the correct answer is here. Answer C. That is called Okazaki fragment. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 19. Assertion: GC-rich DNA melts at a higher temperature. GC base pairs have a free hydrogen bond. Dear students, already we have discussed this question, right? So already you know that 80-rich will be having two hydrogen bonds, two hydrogen bonds. Okay, and GC rich region will be having three hydrogen bonds, right? So the assertion GC rich DNA melts at a higher temperature because here it is more stable, right? More stable, so it needs higher temperature, right? And GC base pairs have three hydrogen bond. Yes, both the assertion and reason will be correct here. So the correct answer A, both true, R explains A. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 20. The length of DNA wrapped around the stone octamer is approximately. So dear student, so if you observe, what is nucleosome? Nucleosome is nothing but the octamer histone protein. Okay, surrounding the histone protein, the DNA is present and it also contain H1 histone altogether. This is called the nucleosome structure, right? And the length of this what? DNA wrapped around the histone is total 146 base pairs. So the correct answer is answer C, that is 146 base pairs are wrapped around the nucleosome here. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 21. The experiment proving semi-conservative replication was performed by. Okay, so dear student, there are many scientist names are there in this chapter. So you should not get confused. But all the scientist names properly. So for they are asking replication process. So the replication process, The semi-conservative replication process was performed and confirmed by the two scientists. Those are Messelson and Stahl. Okay, so the correct answer is here Messelson and Stahl. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 22. The lac operon structural gene that codes for beta galactosidase is. So dear students, so you know that the lac operon is a process. So here the enzymes will involved in breaking of lactose and taking into the cell and involved in the metabolism of what? Lactose. So this is how when the glucose is absent, that time it is utilizes lactose. That is what lac operon process, right? So if you observe here, this is the DNA molecule, and here there are three structural genes, that is LAC-Z, LAC-Y, and LAC-A. Their three genes, structural genes are involving. So LAC-Z is involving in the synthesis of beta galactosidase, and then LAC-Y it is used in the it is involved in the synthesis of lac permease, and then LAC-A is involved in the synthesis of transacetylase. So this beta galactosidase helps to break down lactose, and this lac permease it it will take the lactose into the cell, and then transacetylase a proper function is unknown, still it is involved in the metabolism process. Okay, so this is what the function of this what three structural genes. So the correct answer is here. They are asking which gene is codes for beta galactosidase. So the gene LAC-Z. So the answer C, LAC-Z is involved in the coding of what? The enzyme beta galactosidase. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 23rd. Statement 1. Introns are coding sequences. Statement 2. Exons are removed during the splicing. So dear student, just read the statement properly. Is it true? Introns are coding sequences? Not right. So introns are non-coding sequences, right? So already I've studied about the introns and exons. So exons are what here? Expressed one. That means they are coding one. Okay. So if you observe in the image here, so here these are exons, these are introns. Okay. So here introns are removed. So here introns are removed and exons are spliced here to get what? mRNA. So the both the statement 1 and 2 are here what? False. Okay. So the correct answer is option D. That means statement 1 is also false and statement 2 is also false. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 24. A codon AUG codes for. So dear students, already we have studied the table of genetic code, right? Genetic code. So those are 64, right? Containing 20 amino acids. Okay. So in that you have also studied that UGA and UAA and UAG. These three are what? The stop codons. And then here there is a codon that is called AUG. So that codes for what? Start codon. Start codon. Okay. So the correct answer is here, dear student. So that's code for start codon is coding for what? Methionine. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 25th. Which of the following is not a feature of the genetic code? So dear students, already we have discussed about the genetic code, right? So in the genetic code, already you know that 64 codons are there. So 64 codons and 20 amino acids, right? So for the 64 codons, only 20 amino acids are there. So these 20 amino acids are having multiple codons. Okay. So that's why this nature is called degeneracy. Okay. And then here, this is universal also because this codon is all over the world, that is same. Okay. And that's why it is called universal. And codon are always what? A triplet in nature. That means these are containing three nucleotides. Okay. So that's why they are triplet in nature. And one more is here they have given ambiguous. Is that true? No. So this is not ambiguous. Here these are not creating any confuse. Okay. So there is a very clear clarity here. Okay. There are 20 amino acids. They are coding for the 64 codon. So the answer is C. So these are not ambiguous. Okay. So this is not a feature of the genetic code. So the correct answer is answer C, that is ambiguous. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 26. Which RNA has a clover leaf structure? So if you observe here, so this is the what structure of tRNA, right? So it is the smallest RNA. We have already studied that, right? So here this has clover leaf structure, right? So this structure is called what? Clover leaf structure. So the correct answer is here. Which one, dear student? Option B, that is tRNA has a clover leaf structure. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 27. The central dogma of our molecular biology was given by. So dear student, so first you should understand what you meaning of central dogma. Central dogma is nothing but how the genetic information flows. Genetic information flows. That is what called central dogma of a biology. Okay. So if you observe here image, so the DNA is undergoing the replication process. Okay, the process of making a copy of itself, that is called DNA replication. After that, from the DNA, the process of copying DNA into the mRNA, so that is called, so here we are getting mRNA, that process is called transcription. And then from that, here it is undergoing the translation process to form what, the proteins. Okay, so this is what, how the genetic information flows. Okay, so that is what called central dogma of a biology. So this process is explained by the scientist who is called a crick. Okay, so the correct answer is here, answer C, that is crick. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 28. The genome size of a human approximately. So genome means what? Dear student, our DNA. Okay, so you know that. Let me draw properly. Okay, so this is what, chromosome, right? So chromosome will be containing what, this nucleosome structure, right? Histone protein like that. Okay, so this is how the double stranded DNA. Okay, so this is what the genome, human genome we call. Okay, so here this has approximately 3 into 10 raised to 9 base. Okay, so this is what approximate size of the human genome. So the correct answer is here, option B, raised to 10 raised to 9 base are present in what, the human genome. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 29. If a DNA sample contains 40% of GC, what is the percentage of it? So dear student, it involves a very small calculation. So they have given GC percentage is 40%. So according to Chargaff's rule, so you know that adenine sequences are equals to the thymine and guanine sequences are equals to the cytosine. So they have given GC is what, 40%. So here let's calculate the 80 ratio. Okay. So 100%, right? So here 80 we don't know, but we know the GC percentage, that is what, 40. So for 80 what we will do, we will minus 40 in 100, so we'll get what, 80 percentage now. So 80 is equal to what, 60%. So the correct answer is here, option D, that is what, 60%. So thank you. Hello everyone, Welcome to Sankalp Learning Solution. Let's solve the question 30. A linker stone is. So, dear student, so already I studied the structure of nucleosome, that is containing the octamer structure, that is what the histone protein, that is H2A, H2B, H3 and H4 are present in pairs, right? So that is called octamer, right? So here they are asking which is linker histone. So which is linker histone here? So already I've studied that H1 is a linker histone. Okay. So here it is linking one nucleosome to another, linking another nucleosome. So the linker histone is, the correct answer is option A, that is H1 is linker histone. Thank you. So dear students, are you getting my points? I think all of you are understanding all the questions and answers, right? Okay. So let's, hello everyone. Welcome to the Sankalp Planning Solution. Let's solve the question 31. The RNA world hypothesis suggests that. So, dear students, so according to RNA world hypothesis, so it says that RNA evolved first. RNA evolved first because according to them, RNA has a single stranded structure, single stranded, and it has a self-replicating capability, and also it has a dual function as DNA is performing only one function, that it is storing the genetic material, but here RNA, it can also store the genetic information as well. It is acting as a catalyst. Okay, so that's why according to RNA hypothesis, so it suggests that RNA is evolved first. Okay, so the correct answer, not a DNA evolved and not a protein evolved first, not a carbohydrate evolved first. So according to RNA hypothesis, RNA evolved first. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 32. The enzyme that unwinds the DNA helix. So dear students, already we have discussed the enzymes involved in the replication process, right? So here whatever you are seeing in the picture, that is what the process of what? Replication. Replication. So, which enzyme that unwinds the DNA helix? Okay. So, already you know that there is an enzyme called helicase, which is recognizing the origin of replication, and then what? It unwinds the DNA strands like this. Okay. So, that means it separates the DNA strands. Okay, so the correct answer is DNA helicase. So DNA ligase, it seals and topo topoisomerase. So that is the enzyme that binds the DNA, and the primase that synthesizes the RNA primer. So the DNA helicase that unwinds the strands. Okay, so the correct answer is here option B, that is helicase enzyme. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 33. Which polymerase synthesizes 28 sRNA? So dear students, already you know that RNA polymerase 1, 2 and 3. These three are enzymes are involved in the process called what transcription, right? So transcription in this process, these three enzymes are involved. So already you know that RNA polymerase 1 is synthesizing 28 sRNA and 18S and then 5.8 sRNA. RNA polymerase 2 that is involved in the hnRNA synthesis. Okay, heterogeneous mRNA. And then RNA polymerase 3 that is small nuclear RNA and then 5S RNA. Okay, so these three are involved in synthesis of these three different RNAs. Okay, so here reverse transcriptase is not a correct answer. So that is another mechanism. So here the correct answer is which polymer is synthesizing 28 sRNA. So here the RNA polymerase 1 which is synthesizing 28 sRNA. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question for 34. In a DNA fingerprinting, the proofs used are generally. So dear student, so DNA fingerprinting is a mechanism. Okay, so it is a process. Okay, so here DNA bands are separated. Okay, for the what identification process. So how this DNA bands are separated here means by adding the probes. Okay. So those probes are having giving dark bands. Okay. So we can easily identify now. Okay. So here this what DNA fingerprinting the probes will be containing V and TRS. Okay. So that is called variable tandem repeats. Okay, so these are very unique to the individual. So in the DNA fingerprinting only, so this is we are going to find. Okay, that uniqueness through that we can apply it for so many what processes. Okay, so that's why DNA fingerprinting is useful. Okay, so here the correct answer is here which probes are they are using. So they are using the probes which is called a radio labeled DNA. So the correct answer be radio labeled DNA. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 35. Which DNA form is right-handed? So dear student, so here there are three DNA forms, right? A form, B form and Z form. Actually, this B form is human DNA. If you observe properly, both A form and B form having what? This right-handed helix form. Okay. So the correct answer is here now. What? Answer D, both A and B. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 36. How many turns will a 3400 base pair bDNA molecule have? So it is also very small calculation, dear student. So they are asking for the 3400 base pairs, how many turns it will be having? How many terms will be there? Okay, so you have already studied that for each turn, per turn it is having what? 10 base pairs, right? So now you can easily calculate that 1400 by 10. So 1 340. Okay, so the correct answer is here 340 ton. For the 340 tons, it will become 3400 base space. Okay. So the correct answer is here, dear student. So answer B, that is 340 tons. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 37. Which part of the gene binds transcription factor? So dear student, they are asking during transcription to which part, to which part is act as a initiation for the which process? Transcription process, right? So which part actually here acting? So in the transcription process, what they are doing? They are forming from the DNA, they are forming what? mRNA strand. Okay. So here it is DNA, here mRNA. So during transcription, so it recognizes the promoter sequence. Okay. Promoter sequence. It is called promoter sequence, or it also called enhancer. Okay. So that will initiate the process. The process. Okay. So the correct answer is here. The answer B, that is enhancer. It is also called as a promoter sequence. Okay. Thank you. Hello everyone. Welcome to the Sankal Planning Solution. Let's solve the question 38. During translation, the peptide bond formation is catalyzed by. So dear student, so here they are asking during translation. What is that? Translation is a process of forming the proteins from the mRNA. Okay, so this is what the translation process. So this one is mRNA. Okay, so here this tRNA involved in the what? So this one is polymerase enzyme. Okay, involved in the process of formation of what? The protein amino acids are going to form here. Okay, so here it is forming the amino acids like this. Okay, so that becomes what? Protein. That becomes protein. Okay, so here each amino acid is coding for the particular protein. So this each amino acid is combined or joined by the bond that is called peptide bond. Okay, the bond present between the proteins are called peptide bond. Okay, so that is catalyzed by the enzyme that is called peptidyl transferase. Okay, so this is the enzyme which is involved in the formation of peptide bond during the translation process. Okay, so the correct answer is A, that is peptidyl transferase. Thank you. Hello everyone, welcome to Sankal Planning Solution. Let's solve the question 39. The Shine Dalgarno sequence is found in. So dear student, so whatever you are observing in the image here, that is what this sequences. So the 7 base pair. Okay. So this 7 nucleotide sequences that is called Shine Dalgarno sequence. Actually it is an ribosomal ribosomal RNA binding site. Okay. So here what? So this ribosomal RNA, so which is involved in the translation process. So ribosome which holds the mRNA, right? So that ribosome which holds both mRNA and tRNA. So actually this ribosomal mRNA which attaches to the sequence, that sequence is called Shine-Dalgarno sequence. That means it enables the initiation of the translation. Okay, so the correct answer is here. So it is in case of what? Prokaryotic mRNA. Okay, so signed diagonal sequence is found in only in prokaryotic mRNA. Thank you. Hello everyone. Welcome to Sankal Planning Solution. Let's solve the question 40. The sugar present in the DNA is. Okay. So dear student, so here first you should understand the components of DNA. So already I've studied that the DNA is containing 3 components, right? 3 components. Which are those? One is pentose sugar, right? Another one is what? Phosphate group. Third one is nitrogenous bases. Okay, so if you observe here, this is what the sugar, pentose sugar. This is what the phosphate group, and this is what the nitrogen base. Okay, so all together called it is nucleotide. Okay, so this nucleotide that is present in the DNA. Okay, and this sugar present in the DNA. Which is sugar molecule? The pentose sugar is a sugar molecule. So in case of DNA, deoxyribose is a sugar that is present in case of what? DNA. In case of RNA, that is ribose sugar is present. Okay. So the correct answer is here. Which one? Not a ribose, not a glucose, not a fructose. Here it has deoxyribose sugar. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 41. A nucleosome consists of. So, dear students, already I have studied that nucleosome is nothing but that is containing the histone proteins. That is what? H2A, pair of H2A, a pair of H2B, and a pair of H3 and H4. So, that is what called histone proteins. Okay. So, how many histone proteins are present here? Total 8 histone proteins are present. Okay. Total what? 8 histone, right? So here, whatever present here, that is present in the pair. That is H2A, 2, H2B is 2H4, 2, H3, 4. So total it becomes 8. So the correct answer is here, dear student. Not 6, not 10, not 12. So the answer B is 8 histone proteins are present. Okay. Thank you. Hello everyone, welcome to Sankalp Planning Solution. Let's solve the question 42. Which enzyme seals Okazaki fragments? So dear students, already you have studied that during the replication process, so here it cannot synthesize directly, it cannot synthesize from 3 dash to 5 dash, right? So that's why here what this, what the DNA polymerase 3 will do, it adds the small fragments. Okay, so that fragments are called Okazaki fragment. Once the synthesis is completed, so here it becomes what? Lagging strand, because here it has very small fragment. If you observe here, this is one fragment, this is one fragment. So after process, it has to be sealed in the middle, right? So that sealing process is done by the enzyme that is called DNA ligase. Okay, so the correct answer is here what? The enzyme ligase, option A, that is involved in the process of sealing the Okazaki fragments. Thank you. Hello everyone. Welcome to Sankalp Planning Solution. Let's solve the question 43. Template strand is also known as. So dear student, so already you have studied that template strand in case of replication we have studied, right? So template strand is also known as non-coding strand, right? So according to the non-coding strand, complementary strand will be synthesized. So that is what called coding strand. So a non-coding strand is also called as antisense strand. Okay. So if you observe here, so template strand is called anti-strand. So according to anti-strand, sense strand is synthesized. That means complementary strand become sense strand. Okay. So thank you. Sorry. The option, which one is correct here, dear student? So the option that is antisense strand is correct. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Which RNA is involved in the splicing? So what do you mean by splicing, dear student? So removing the introns, that is what? Non-coding sequences, non-coding region, and adding the exons, sealing the exons. That is called what? Splicing mechanism, right? So here, which RNA is involved? They are asking. So there are three enzymes: RNA polymerase 1, 2, 3. Right? Already I have studied that RNA polymerase 3 is synthesizing snRNA, and then what? Another one 5S RNA. Right? So here small nuclear RNA. Okay, so that is involved in splicing mechanism. Okay, so the correct answer is here option B, that is small nuclear RNA that is involved in the splicing mechanism. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 45. Assertion. A mRNA carries codon. Reason, tRNA carries anticodon. See, dear student, mRNA is carrying codons. Yes, yes. So mRNA is always carrying the codon like this, because it is involved in the what process? Translation process, right? So tRNA carries anticodon. Yes, tRNA is carrying the anticodon. Yes, anticodon site is present in the tRNA, right? So this is what the structure of tRNA. Here there is what? Anticodon site is there, right? So that is also correct. But here read that here they have given assertion and reason. Is that true? This is the reason for what mRNA carrying the codon. No, right? So here the correct option is the option B, both true only, but R is doesn't explain A. R is not explanation for the A. But both the sentences are correct here. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 46. Okay. The first step of translation is, so dear student, what do you mean by translation? Synthesis of the protein from the mRNA to protein. That process is called what? Translation, right? So here if you observe, so there are three main steps involved in the translation. Initiation, that means here the translation process is initiated. That means here the ribosomal RNA that binds to the mRNA and starts the process that is called initiation. And next process is called elongation. Here what happens? Here it synthesizes. That means here the tRNA is coding for the mRNA, and it is giving the amino acid. So that is what we call elongation process. And there is last process, that is termination. Here it will meet the stop codon and it stops the process of translation. In the end, we are getting the protein chain. Okay, so this is what the process of what translation, that is synthesis of protein from the mRNA strand, right? So here the first step is what, the initiation process. So the correct answer is here, dear student, option C, that is what, initiation process. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 47. The lack operon is an example of. So dear student, so already you know that lack operon is a process what? So here it is a process of breakdowning lactose and lactose metabolism, right? For the energy source. So if you observe, so when this lac operon is gets activated, when it gets activated, when the glucose is absent and when the lactose is present. Okay, so here lactose is acting as a inducer. Okay, so that's why here lac operon is an, it is called inducible operon, because here only in the inducible what lactose, this lac operon process is gets activated. So that's why this process is called what, inducible operon. So the correct answer B, that is inducible operon. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 48. VNTRS are used in. So, dear students, already you have studied that. You have studied and you have a little knowledge about the DNA fingerprinting, right? DNA fingerprinting. Okay, by using this uniqueness in the every human being, every human, every human. So, DNA fingerprinting is useful. So here variable tandem repeats, variable number of tandem repeats. Okay, so these are the sequences, same coding sequences that is present, that is very unique to the individuals. Okay, so that's why it is very unique, right? So that's why it is applicable, it is used in the process that is called DNA fingerprinting. So already you all know that DNA fingerprinting has so many application to find out the culprit and to find out the parents of the unknown what uh, child and all. So here that is what called parental dispute. So here in all these what uh, 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 what in all these what areas in all these DNA fingerprinting is used. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 49. A ribosome is. So dear student, so you have studied the three types of RNA, right? So one is mRNA, that is called messenger RNA, rRNA, that is called ribosomal RNA, and tRNA, that is called transfer RNA, right? So already I have studied three types of RNA. So here ribosome, that belongs to the what? rRNA here, because here this is a part of rRNA. Okay, so that is called ribosomal RNA. So in case of prokaryotic, it is called 70S ribosome sediment here, and here Sedberg unit it is called, and here in case of eukaryotic, it is called 80S ribosome. So yes, the larger fragment will give 50S and the smaller subunit will be 30S in case of prokaryotic. In case of eukaryotic, a larger subunit will be 60S and 40S will be a smaller subunit. Thank you. So sorry. So the correct answer is here what? The ribosome is an RNA enzyme. Okay. So the option B, that is RNA enzyme, is an correct answer. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 50. A major and minor grooves are characteristic features of. So dear students, already have studied the structure of BDNA, that is human DNA, right? So clearly you know the structure of BDNA, that is for each turn it is 3.4 nanometer what in diameter, and then here it has the adjacent base pair will be having the distance 0.34 nanometer, and then 20 angstrom will be the length of the DNA, sorry, diameter of the DNA. And then here, if you observe, it contains two grooves. Okay, so here this is called major groove, and here it is called minor groove. So both the grooves are present in each turn. Each turn. Each turn will be containing both major and minor grooves. Okay. So here this is the characteristic feature of what? BDNA. So the correct answer B, that is BDNA. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 51. Which of the following is not found in RNA? So dear students, so already you have learnt the components present in the DNA, right? In case of DNA pentose, sorry, deoxyribose is present, right? And so here only I will explain. So the only one difference, two differences are there. In case of DNA, that is containing deoxyribose nucleic acid, and in case of RNA, ribonucleic acid is present, right? And another difference is here in nucleotides. See here Three nucleotides are similar in both DNA and RNA. That is adenine, cytosine, guanine. Okay. And in case of RNA, thymine is replaced by the uracil. Okay. So in the DNA, we'll find the thymine nucleotide. In case of RNA, we'll find the uracil. Okay. So that's the major difference in RNA and the DNA structure. Okay. So the correct answer is here, which is not found in the RNA. So here, which is not found? Not found means here the thymine which is not present in the RNA, right? The phosphate is present, even ribose is present, and even uracil is present. Only the thymine is absent here. Okay. So thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 52. DNA fingerprinting was developed by. So, dear student, so already I've studied little about the DNA fingerprinting, right? So here this process was developed by uh, the scientist who is called Alec Jeffreys. Okay, he invented this process in 1984. So the correct answer is here not Harvey, not a uh, Nirenberg, not a Boyer. So the correct answer is here Jeffreys. Okay, so Alec Jeffreys he found the method of what? The DNA fingerprinting. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 53. Which bond connects the nitrogenous base to the sugar? So dear students, so already we have discussed about the bonds present in the DNA also. In while explaining the first question only, I explained very clearly. So, in between the phosphate and the sugar, there is a bond that is called ester bond. So, it is called phosphodiester bond. Okay, phosphodiester bond. Okay, and the bond present between the nitrogenous bases and the sugar. Okay, so here whatever the bond that is present between the sugar and nitrogen bases, because here to the ninth position of the pyrimidine, purine, here it is ninth position. So here one hydrogen is attached. So to the ninth position here, the sugar molecules first position gets attaches and it forms the bond. N means here N is present ninth position. So N. And glycosidic means what? Glyco means sugar. So that's why glycosidic. Glycosidic bond. Okay. So the correct answer is here. The bond which is present between the nitrogen base and the sugar is that is called glycosidic bond, or it also called N-glycosidic bond. Okay. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 54. Chromatin that is actively transcribed is. So dear student, the chromatin structure, so in the chromosome there will be a two regions. Okay, so here if you observe orange colored region, Okay, so that is called heterochromatin. Okay, and the blue color region, that is called euchromatin. Okay, so here euchromatins are loosely packed. Okay, so here these are loosely packed. So here this euchromatin region is actively participating in the what, transcription process. Okay, but this orange color region is very tightly linked, tightly packed. Here it is. So that's why here this region, orange colored heterochromatin region, is not actively participating in the transcription process. Okay, so the correct answer is here, the chromatin that actively transcribed is euchromatin region. Okay, thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 55. Which enzyme synthesizes the RNA primer? Okay. So, dear students, so RNA primer, you know that directly the DNA polymerase 2 cannot add the nucleotides in the replication process. So, here during for the initiation, it adds the RNA primers, right? So, that RNA primer is synthesized by the enzyme that is called primase. If you observe here, primase is an enzyme, so that is synthesizing the RNA primer. Okay, so this is the correct answer. So the answer, that is C, that is primase is an enzyme which is synthesizing the RNA primer. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the question 56. The promoter region in prokaryotes includes. So dear student, in case of what, the transcription process, we have already discussed that the there will be a promoter region, that is also called as enhancer, right? So the enhancer region or promoter region, that should contain the sequences, so where exactly the process will get start. Okay, so in case of prokaryotic, there will be a two region, that is called minus 35 region and minus 10 region, that will be containing the sequence. So six base pairs here, TTGACA, TT TAT. So these are the two regions present in the prokaryotic. So that is called the promoter region. Okay. So the correct answer is in case of prokaryotic. So minus 10 and minus 35 sequences that acts as a promoter region. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 57. During translation, the ribosome moves on the mRNA in. So, dear students, you have studied that the it can synthesize the strands only from 5 dash to 3 dash. Okay, not 3 dash to 5 dash direction, right? It cannot synthesize from 3 dash to 5 dash, and in random direction, that is also not correct, and opposite direction alternately, that is also not correct. So the correct answer is here, if you observe properly here, from 5 dash to 3 dash, the process is ongoing, right? So the correct answer is here option B, that is 5 dash to 3 dash direction. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 8. During splicing, the removed part is called. So dear students, splicing is a mechanism, so it involves removal of what introns and the sealing of what the exons, right? So already I've studied that introns are what non-coding sequences. These are non-coding sequences, right? So these are removed during the splicing. And if you observe here, two exons are added here. Okay, spliced exons and splicing intermediate. So here they are removing, and here it is a pre-mRNA. Pre-mRNA will be containing the introns. So here we have to remove this intron, that is non-coding sequence. So here removal of what? So removal of introns. That is the correct answer. So answer B is the correct answer. Thank you. Hello everyone. Welcome to Sankalp Learning Solution. Let's solve the question 59. A reverse transcriptase is found in. So dear students, if you observe the structure here, so here you know the process, that is what the reverse transcription, right? Transcription you know, right? So what is that transcription? Synthesis of mRNA, right, from the DNA, right? So here reverse means what? It is a dead opposite process, right? So that means here from the RNA, here it synthesizes the DNA. So here this is possible only in case of some of the viruses. So that is called retroviruses. So retroviruses RNA has a capability that is called reverse transcriptase. So reverse transcriptase enzyme is present. So that is involved in the process of reverse transcription. So here it synthesizes DNA from the RNA. Okay. So that is in case of what? Retroviruses. So the correct answer is reverse transcriptase mechanism found in retroviruses. Okay. Thank you. Hello everyone, welcome to Sankalp Learning Solution. Let's solve the last question of the molecular basis of inheritance, that is 60. Which is the smallest RNA? So dear students, you can answer, I think, because so many times I have told you while explaining the structure of tRNA. So it is the smallest RNA. Okay, so mRNA is also longer one, and uh, ribosomal RNA also uh, a little bigger one compared to a tRNA. Okay, so tRNA is the smallest one, containing a single strand which looks like this clover leaf, and it is folded like this clover leaf. So that is considered as what? A very smallest RNA. Okay, so the correct answer is here option C, that is tRNA is a very smallest RNA. Thank you. So we'll discuss now what are the summary of the today's class. So we have discussed the structure of the questions of DNA and the packaging of DNA helix, right? And we also discussed the questions on the genetic material and the properties of the genetic material, right? And also discussed the RNA world and the process of replication. And we also discussed the questions on transcription, right? So many questions we have discussed on the topic of on the transcription. And then we also discussed the types of RNA, that is what rRNA, tRNA, mRNA, and the process of transcription also we have discussed. And we also discussed the genetic code table. Yes, we have discussed the questions on that 64 codons, triplet codons, and then 20 amino acids. And then we also discussed the questions on tRNA translation process, and then regulation of gene expression, that is what lac operon process also we have discussed. And we also discussed the human genome project and the DNA fingerprinting. Yes, so these are the topics we have covered in today's session. Thank you.